everyone, I'm Dr. Nicole King. I'm so excited for today's activity. I hope you are too. We will be exploring animal cells. As you all know, living things are made up of cells. Within cells, organelles have specific duties and jobs that they must complete. There are two types of cells, prokaryotic cells and eukaryotic cells. Prokaryotic cells are simple, and these cells are typically bacteria and other unicellular organisms have prokaryotic cells, whereas plants and animals have eukaryotic cells. Eukaryotic cells are more complex than prokaryotic cells, and they have a lot of different organelles and a lot of different parts. So we will learn about this in today's lab. Animal cells are specialized, and they have a lot of different jobs and a lot of different duties. Cell biology is the study of cells and their processes. When a cell biologist studies cancer research, they learn about the different processes of cell division as well as cell transformation from healthy cells to cancerous cells. Let's get ready to go into the lab. All right, look forward to seeing you there with me. Thank you all for coming with me and joining me in the lab. So now we're going to go over our supplies before we do our demonstration. So for our lab today, we're going to need a plate, which will represent our cell, a nice protective covering so we can make sure our space is clean and we don't mess up anything, paper towels, a spoon, we're going to need some whipped cream, we're going to need some red icing, and we're going to need a lot of different candies. And just to let you all know, this is one of our labs that we will be able to eat. All right, so the first organelle we're going to build is the cell membrane. So the cell membrane is semi-permeable and it serves as a protective barrier. So we're going to need our red icing. And what we're going to do is we're going to take our red icing and we're just going to build a barrier along the plate. So along the lines of the plate. All right. Nice and easy, smooth sailing. Look at our cell membrane. Next organelle will be the cytoplasm. We will use whipped cream to represent our cytoplasm. So the cytoplasm is a jelly-like substance which fills the cell. So once we add our cytoplasm, we're going to use a spoon to kind of smear it out. And within the cytoplasm are enzymes. And enzymes, we know, act as biological catalysts. They help to speed up reactions or chemical reactions. Our next organelle we will add is the nucleus. The nucleus carries and regulates the functions of the cell. It also houses DNA, and as you all know, DNA stands for deoxyribonucleic acid. We're going to use our rheses to represent our nucleus, which we will add to our cell. So as we open the rheses, we're just going to need one. And we're just going to sit it on top of the cytoplasm. Our next organelle we will add will be the nucleoli. The nucleoli serve to manufacture ribosomes, which we will get to a little bit later. The nucleoli sits inside the nucleus, but for our cell, we'll either add it on top of our nucleus or if you would like to add it inside of our nucleus. And remember, our Reese's peanut butter cup is our nucleus, sorry about that, within our cell. So I'll go ahead and take a gumball and I'm going to add it just on top. We just need one gumball. Well, actually, you know what? I'm going to fit it on the inside. Our next organelle, which we will add to our cell, will be the ribosomes. The ribosomes are free-floating within the cell membrane, and they are where proteins are made. So we're going to use our nerds, and we're going to add them to our cytoplasm. And we can take a few nerds and just kind of spread them out. Our next organelle we will add to our cell will be lysosomes. Lysosomes help to break down old or worn out parts within the cell. We will use M&Ms to represent lysosomes in our cell. So I'm going to open up our M&Ms and take about 2 to 3 of them and just kind of spread them out. Next up, we have mitochondria, which is the powerhouse of the cell. Within the mitochondria, that's where aerobic respiration takes place. We're going to use our raisins to represent the mitochondria in our cell. We're going to take about 2 to 3 raisins and spread them out. Our next organelle we will add to our cell will be the endoplasmic reticulum, also known as the ER. The ER are tubes within the cell, and they make and transport proteins and fats. They also work with ribosomes, which serve as the rough ER in the cell. So we're going to take some Twizzlers, and we're also going to add our nerds to represent the ribosomes. So let's open up our Twizzlers, and we're going to break them into 2 to 3 pieces. You just need 1 Twizzler. And then we're going to get our nerds that we used previously to kind of put right next to it. And since the ER transports proteins and fats in and out of the cell, we're going to make sure that it's close to our barrier. And we're going to add some of our nerds from before. So our next organelle we will discuss will be vacuoles. And vacuoles help to isolate waste and waste products. We're going to use our lifesavers to represent vacuoles in our cell. So vacuoles also help to maintain water balance within the cell. Well, everyone, we just finished making our cell. Everyone did a great job. So this is what mine looks like. Hopefully you all can see that. And so for one last time, we're just going to go over the different parts of a cell. We have the cell membrane, the cytoplasm, the nucleus, the nucleolus, ribosomes, lysosomes, mitochondria, vacuoles, endoplasmic reticulum, and the Golgi apparatus. Something else we can do is look at cells underneath a microscope. So I have my microscope here and I have some slides here. So let's go take a look at what some of these slides look like under a microscope.
Hi everyone. We are looking at a cell under a microscope. We're currently looking at a human liver cell. And if we look closely at our observations and think about this, we notice that the color is red. It looks very healthy. This cell looks very normal. Now let's take a closer look at this cell I just added under the microscope. This is a human liver cell. However, this is a cancerous cell. And you can notice some differences in the cell shape and in the cell color from the previous healthy human liver cell that we looked at under the microscope.
I hope you all enjoyed today's lab. I had a great time learning about animal cells, cell biology, and the basics of cancer research. As we always like to close out our labs, we like to just shout out STEAM forward. So count down with me. 1, 2, 3, STEAM forward! Till next time, bye.